Good evening, everyone. My name is Jayne Miller, and I am happy to be your guest moderator tonight for Johns Hopkins Medicine, A Woman's Journey, Healthy Conversations That Matter webinar. On behalf of Johns Hopkins Medicine's A Woman's Journey, thank you for joining me this evening for A Woman's Journey's monthly webcast series, Healthy Conversations That Matter. A Woman's Journey continues to strive to improve well-being through health education. The American Cancer Society estimates that the U.S. will see over 2 million new cancer cases in 2024 and more than 611,000 cancer deaths. Tonight, I am joined by Johns Hopkins oncologist and director of the Sidney Kimmel Comprehensive Cancer Center, William G. Nelson. Please use the Q&A on your screen to pose your questions to Dr. Nelson, who will respond during the last 20 minutes of our program. Tonight's webcast will conclude at 8 p.m. A Woman's Journey would also like to acknowledge and thank Johns Hopkins University's program, Hopkins at Home, for their production assistance. You can visit their website at https://hopkinsathome.jhu.edu for additional lectures and courses throughout the year. A Woman's Journey acknowledges the support of the Wiglicki Family Foundation, sponsor of Healthy Conversations That Matter. And now, I am pleased to welcome Dr. Nelson.
Jayne, thank you very much, and it's wonderful to be with all of you. As Jayne Miller mentioned at the beginning of her remarks, this year something on the order of 2 million people will hear the 3 words, "You have cancer," that will upend their lives, forever change the way they interact with their family, their friends. This is why we're here, right? The good news is over the last 3 decades, there have been extraordinary progress against cancer. Truly remarkable that fills people like me with a lot of optimism and enthusiasm for where we'll go in the future. For instance, of the 2 million people diagnosed with cancer this year, at least 2/3 of them will live 5 years or more. And there's a growing collection of people in our society, 18 to 20 million probably, who are cancer survivors. Sounds like they should be a political party in some kind of movement. There's a great estimate that since 1991, we've averted at least 4 million cancer deaths. So we're beginning to make progress. Why are we making so much, and what's going to happen in the future is what we're going to dive into a little bit tonight. So first, how long have we known about cancer? And there's a delightful book, if you have a chance to read it, by Sid Mukherjee called The Emperor of All Maladies. Sid's a leukemia doctor at Columbia, actually, and he wrote this piece on the history of cancer. And he charts back the human understanding that cancer exists to Imhotep. Imhotep was the basis for the Mummy and the Mummy movies, right? Boris Karloff and I think more recently Arnold Vosloo played the Mummy. Apparently, he was quite an important counselor to the pharaoh, and he was a religious figure. The key, though, is that he wrote down things and people wrote down things later that he described. And so the Edwin Smith Papyrus, which was discovered in the 1800s, has a description from Imhotep about breast tumors. So breast cancer, at least 3,000 almost years ago. He compared the field of breast tumors to the unripe hemat fruit, which is hard and cool to the touch. I have no idea what a hemat fruit is. I've actually tried to figure out, I think it's some kind of mango or something. Nonetheless, apparently it was hard and cool to the touch. And what he did say at the time was, as for therapy, there was none. And so we've come a long way since then, thank heavens. And the reason is because all cancers are fundamentally acquired disorders, disorders of acquired defects, if you will, in genes and in gene function. So we have to think a little bit about what genes are and what the genome is. So here's the small part of the biology lesson. You get to ask me questions. I don't get to ask you test questions at the end, so don't worry about this. The human genome has 3 billion base pairs, A, T, G, and C, that you know about. What's interesting about that number is that if you take those as letters and you say how many Encyclopedia Britannicas would this fill up, it would fill up something like 20 full copies in one cell in one person. 6 feet of DNA wrapped up and stuck into something that's the size of 1/1000th of an inch or smaller. What's interesting is if you look at your neighbors, look at your friends, look at your folks you work with and whatnot, you realize that 99.9% identical you are to everyone you meet. Human beings are quite similar to each other in terms of their genetic code. But that means that 1 in every 1,000 base pairs are different. Every one is actually unique, except for identical twins, and they become unique over time. That means there's at least 3 million differences between any two individuals, and that's before any acquired changes in the gene. So what you might already be wondering is that every cancer that develops in any person is going to be unique. It's not going to be exactly like any other cancer formed in any other person. So the 6 feet of DNA stuffed into a nucleus in a cell that's less than 1/1000th of an inch is packaged in there in such a way that certain genes are more usable than others, and this differs from cell to cell. As you can imagine, the cells in the retina of your eye might use different genes. The genes are the blueprint for all the components of the cell, so they might use different blueprint to make specialized things for the retina in your eye that would be different than the cells in your colon, the cells in your breast or prostate or lung. A typical cancer cell has acquired 3,000 mutations, we call them. That's a change in the ATGC sequence. More than 100 rearrangements, that's where the DNA has been broken and plugged back into the wrong place. And at least 5,000 or more changes in the way the DNA is packaged, marked by these tags on the DNA called DNA methylation marks. So that's a lot of acquired defects. And what they do is they are the changes in the things that drive the cells to be as nefarious as they are, to proliferate in an uncontrollable way, so they outgrow their normal environment, to invade through normal tissues and disrupt them, to travel to distant sites in the body we call metastasis, to travel to a different site, grow in those sites and disrupt those functions, and of course, ultimately, many times to threaten life. We recognize 2 classes in the sense of gene defects. One we're called oncogene, that quite literally means a cancer gene, which when it's mutated to an active form, it becomes a driver. It's sort of like an accelerator that's stuck in the on, the press down position. The other are tumor suppressor genes. These are genes when they're damaged or broken and don't create any protein products, would have otherwise been the brake. So you've lost the brakes of the cell. So both of these occur. Many of each occur in a typical cancer cell. And what they have is a normal cell will acquire a defect, become a cancer. They may acquire even more defects over time, develop the ability to travel throughout the body. This is the logic for why early detection might be helpful. You want to get at the cancer before these kinds of things have happened. And then some of our best treatments historically, which might kill a lot of the cells, there might be one acquire a new defect that can have it outfox the treatment and this kind of thing. So as you might guess, if there's that many difficulties or that many defects in the genome of the cancer cells, what about gene sequencing? If we can use gene sequencing to find one hair follicle at the crime scene in a Law and Order episode, can't we use the same kind of technology to find a cancer cell wherever it might be, or its contents wherever it might be? And the answer to that is yes. At this point, we can sequence anybody's cancer genome pretty quickly at what's an increasingly lower and lower costs. And we're beginning to use this for all kinds of things. You could imagine you might use it for cancer screening. Can you detect a cancer somewhere before you can see it on an image or feel it in your body? Early detection might follow that. Can it help with a cancer diagnosis to say these defects might be more associated with breast cancer than colon cancer, that be more confident that that's a cancer? Can you characterize how nasty the cancer is going to behave? Is this the kind of thing that surgery alone can treat effectively, or will more treatment be needed? Can you use the sequence information to generate those kinds of impressions? And can you classify the cancer? Increasingly, this is very common with a particular set of defects. Was that a defect in the cancer that would respond to one treatment but not another? And this is where you're beginning to individualize the cancer treatments. In one woman's breast cancer may have different defects than another, may respond to a different treatment than another. Now, this is not surprising. We had known for a long time that certain women's breast cancer responded to blocking the action of the female hormone estrogen. Other women's cancers did not. So we already knew that this was an issue, but now we have a detailed inventory of all the defective genes. We begin to be ever more precise in the way we can construct and deliver treatments. You might wonder, for early detection, where does cancer DNA go? Where can we look for it to detect? It looks like when cancers arise in the surface type lining cells, and this is most cancers. This is skin cancer, cancer in the mouth, the esophagus, the stomach, the colon, The breast is effectively a surface thing, the prostate, all these kinds, the lung, et cetera. All these will shed, even when they're precancerous, will shed cells and their DNA into wherever it's going. If it's a bladder, it'll shed into the urine. If it's in the lung, it'll shed into the sputum. If it's in the mouth, it'll shed into the saliva. And there are many approaches to take these bodily fluids and find DNA in them. One of them is already done somewhat, is sort of a more modern version of the pap smear, where you can swab the uterine cervix. You can do this yourself even, and look for the DNA that's in a virus called human papillomavirus, which forms, it accomplishes the function of one of the defective genes in many other cancers, and you can detect the cancers that way, or in a precancerous phase, it's easier to treat. Once the cancer invades into the tissue, the cells end up in the lymphatics, they end up in the circulation, and the DNA can be detected in a blood test. And this is something that has just exploded in the last 5 or 6 years. Called circulating tumor DNA. This is the DNA that circulates from the cancer that's invaded into a tissue somewhere. And you can imagine in a graph like this, if you look at the levels of this, you could imagine that it begin to rise. You might be able to detect it for early detection, detecting it earlier than you could have waiting for an image or waiting for you to feel a lump or what have you. You could imagine a treatment, if it was effective, then all that cancer DNA should go away. If it wasn't completely effective, even though it looked effective and there was still cancer DNA around, it had to come from somewhere. You might imagine that is coming from cancers that persisted, and that might be the right person to use additional treatment, which some people call adjuvant therapy, additional treatment. If there isn't any cancer DNA, you could imagine not needing to use adjuvant therapy. You certainly will get an early look if the cancer is coming back, be able to treat it in a much earlier phase when the person's healthy, and you can use it to monitor treatment. All of these things are occurring. Many companies started to try and do this as well. One of the ones that came right out of here from Johns Hopkins started a company, some of the folks from here, that went on and was ultimately purchased by Exact Sciences, or the Cologuard people. And that was a study they did using the Geisinger Clinic people up in central Pennsylvania, where they looked at 10,000 women who were not known to have any cancer. They all had a blood test, and then they all also underwent the screening kind of studies that you would normally do for cancer at their age, mammography, colonoscopy, and the like. And what they found were 26 cancers were detected by the blood test, another 24 by the routine screen, 46 by other means. So there were a significant fraction of cancers that could be detected only via this blood test. Now, this is just the first attempt to do this. This has to be refined, figure out who it's going to help, and this kind of thing. But this is what I think we're looking at in the future: DNA testing for cancers, leading to much, much earlier detection. The other benefit of early detection is that we may be able to increasingly use less and less invasive surgical procedures and other things to deal with these tiny cancers. You can imagine at a time before mammography, breast cancers tend to be recognized when they could be felt by a woman and by a physical examination by her doctor. And then to remove that tumor, it was often necessary to remove the entire breast. Once a screening test came, which is mammography, that sees very tiny changes, that often can be dealt with a lumpectomy, preserving the contour of the breast and just a less deforming, a less morbid surgery. You can imagine that all throughout the body, when you begin to detect cancers at ever smaller states, can you use these more modern approaches and less morbid, less deforming kind of procedures to deal with these cancers? Now, treating cancers with drugs has been around for a while. If you look from about 1942 to 2000 or so, most of the anti-cancer drugs we found were found by screening things, looking in the plant world, in the Amazon rainforest, everywhere you can think of, famously in a sewer in Italy. And these are drugs principally. The screening tactic was, could they kill cancer cells growing on a dish in a laboratory? And if they could, typically they all worked about the same way. The more you put on the dishes, the more cancer cells you could kill in the dish. The clinical exercise was, could you get enough of this stuff into a person to kill the cancer cells in their body without doing too much collateral damage? And so the clinical trials were very difficult, right? take folks with advanced cancers for which there was no other treatment, but they were generally healthy, meaning their liver and their kidney worked so they could metabolize the drugs if they were to get into any problems. Then you'd give the first 3 people a very, very low dose of the drug, see what the side effects were, and you'd increase the dose sequentially until you got to the maximally tolerated dose. And that's just what it sounds like it is. It's the maximum anyone can tolerate. And at that dose, you then go out and treat 20 to 50 women with breast cancer, 20 to 50 men with prostate cancer, 20 to 50 men and women with colorectal cancer, and see if it seemed to work in any of those diseases. If it did, then the next question you would ask is it better than the best we have already? Or if we add it to what we have now, is there any improvement? This process of going through these phases of clinical trials would take about 15 years or so. The pharma industry estimates it costs more than $1 billion per approved drug. And the reason for that was that 90% of the ones that were given to people never found a use, never secured FDA approval. Most of them failed to get FDA approval pretty late on in the process. And so lots of invested funds in things that didn't work very well. Having said that, this era, we cured Hopkins disease, cured leukemia in children, the like. It was the beginning of what we were doing. Now everything has changed. In addition to being able to sequence the DNA that I mentioned, the world of chemistry in making drugs is also evolved to a stunning level. It used to be some of these drugs that they would get from plants would have a very complicated chemical structure. You could draw it, but no one could make it. Only the plant could make it. Famously, the drug paclitaxel, which was marketed under the name Taxol, used to be in the bark of the Pacific yew tree. That used to be the only way you could get the drug. What's happened since that time is pretty much now anything you can draw as a chemical structure, somebody will figure out a way to make. So medicinal chemistry has advanced to that point. Now what's happened is the artificial intelligence you've heard so much about, and some of the structural biology that we've been able to do, that's to take these protein components, some of them the products of defective genes, so they're defective components. We can get down to the atomic resolution, exactly where every amino acid is in that protein, where it is in space. As we're doing this, we're learning that all these proteins are little mini machines. They move around to accomplish their functions. And very often we can tuck a chemical of some kind, a small chemical entity, into a nook or cranny in one of these proteins and stop it from working. So if there's a defective cancer gene that's an on switch for the cancer, we can often find a drug to park in that thing to stop it from being an on switch. And these are the kinds of drugs that we are building through this kind of process using artificial intelligence. Remember, a lot of pattern recognition goes on with artificial intelligence to help us do this. And this will only get better and faster. The paradigm, of course, that this is working by and fueling is one that it's a more modern, probably about 30 to 35 years old. The first gene defect discovered in any cancer was discovered by this character named Peter Knoll. And you can see it was a time when the looking at the genomes could only be done by what are called karyotypes. Those are each of the human chromosomes, right? 2 chromosomes, 1, 2, 3, over. And this has 2 X chromosomes, so this was a female, right? And what you can see is he noticed in leukemia cells that chromosome 22, one of the 2 of them seemed a little bit short. It was a little bit funny looking. And when he looked more closely, it turns out that that little area on chromosome 9 was a little bit long. And one of the great women in the history of science, Janet Rowley, recognized that this was a piece of chromosome 22 that had broken and moved over and glued itself into chromosome 9, creating a defective gene as a result of that breakage and refusion, the gene they called BCR-ABL. BCR-ABL, the product of this gene, the component that it's the blueprint for, is an enzyme. And that enzyme, it was an attractive notion 30 years ago because enzymes were thought to be things that were more easy to design drugs to stop. It's sort of like that old movie with Arnold Schwarzenegger called Predator, where they're in desperate circumstances, but they see some green goo on a tree. And I can't do his accent any justice, but Arnold Schwarzenegger says, if it bleeds, we can kill it. Well, if it's an enzyme, you can figure out a way to stop it. And these two characters here, Charles Sawyers is my roommate in medical school and a wonderful person, and Brian Drucker, another close friend. They were sure that we should be able to make a drug to stop this enzyme. And there's a lot of people who were skeptical at the time, but they proved to be right. And what the clinical trial they did was totally different than that strategy I was telling you about before. They didn't want to give the maximally tolerated dose. They just wanted to give enough of the drug to stop this enzyme from working. And they had a way to measure that. They weren't going to treat everyone with every cancer. They were only going to treat people with this kind of leukemia and this specific gene defect. The reasoning was that if they stopped the enzyme from working in this particular collection of folks and there wasn't a benefit, then you would never hear of the drug again. You wouldn't go test it in breast cancer and colon cancer and whatnot. You'd be done. Turns out it worked incredibly well. As you can see, the dose down in that first column, as it gets up to the dose that stops the enzyme from working, everybody responded. Most of the people got complete responses. And based on this very first trial, the FDA approved this drug, and it's still in use for this condition today. Having said that, if you look at the inverse of what they saw, in other words, if it hadn't worked after all this, there wouldn't have been any more money spent on it. What this did was this was an invitation to the large biopharmaceutical companies to get in the anti-cancer drug discovery game. They really hadn't been in it before. But now they had what they thought was that they call a de-risked pathway. In other words, we're going to design a drug for a specific gene defect. We're going to treat people with that defect. If it doesn't work, we're done. And we're not going to spend a lot of money on stuff that doesn't work. We're going to instead move to the next target and find something that does work. The other thing that's happened is protein engineering. In the same way I mentioned that it's extraordinarily what medicinal chemists can do now, it's also extraordinary what you can do with protein engineering. Back in the 1970s, Kohler and Milstein discovered you could figure out a way to make one particular antibody and purify it in such a way that you could give it to people as a drug. There are a number of antibody drugs out there now. The first anti-cancer antibody drug was called trastuzumab. It's against something called HER2 on breast cancer cells. But since that time, the ability to craft these antibodies and make them into just about anything you want has also become extraordinary. One to watch is this one down here, E, where they take the antibody and they link a drug to it. Now you can link a very toxic drug. When it's connected to the antibody and it's infused into your bloodstream, it swims around your body, doesn't do anything. When it gets to the cancer, ideally it gets clipped off right there, kills the cancer cells, but very little else is exposed to it. And there are some incredible success stories already. And today there are 90 separate antibody drug conjugates, they're called, 90 of them in clinical trials for cancer just today. So this has exploded, and we're looking at, by these new chemical entity kind of drugs, ones that can be in pills and infusion, these new antibody-like drugs, you're looking at a completely different set of treatments for cancer that have much less in the way of untoward side effects, and they're destined to be much more effective. And what's happened is there has been an absolute explosion in anti-cancer drug development. This is not intended for anybody to read, especially not with my eyesight. But these are just the drugs approved since 2000 for cancer. It's insane. There's like 78 approvals last year. It is exploding. It's really taken on almost all cancers. And this is why we're starting to see this incredible progress. The other thing that's happened, and none too soon, is that we finally are bringing the immune system into play to tackle the cancer. I think everyone believed the immune system should play a role. We know it plays a role in fighting off viruses like the COVID virus and bacterial infections and the like. We know that vaccinating against things prevents illnesses as you go. That's an immune response kind of effect. And you know, there's a long history of immunotherapy. It dates back to this character, William Coley, who was a surgeon in New York. And he made this sort of witch's brew of bacteria that he'd heated so that they wouldn't reproduce. And he infused them. And what it created was that same kind of what they call cytokine release syndrome that was what went wrong with some people with COVID-19. They got very sick. He basically made them that kind of sick, septic sick. And if they lived through it, then some of their cancer shrunk. And so amazing thing about me is that little bottle was from Park Davis, and they were selling this over the counter, which was an amazing thought to me until they eventually made too many people sick and they stopped doing it. But the question was always, the immune system is incredibly remarkable, right? It can tell the difference between my cells and your cells, which is why if you transplant a kidney into me, I might reject it. It can tell the difference between my cells and a bacteria or a parasite or a virus, of course. It can tell the difference between my cell and my cell that has a virus in it, and for the purpose of destroying the cells that are infected. The question was always, can the immune system see the cancer? Can it see the cancer as different in a way that it could potentially destroy it? And I think at long last, it's very clear that the immune system can and does see the cancer as different. The key is that the cancer was holding the immune system at bay, holding it in check. This is a very complicated slide. There is no exam question on this at all, so put that out of your mind. Basically, the way that the significant fraction, probably about 15% of human cancers, hold the immune system at bay is, I raised adolescent daughters, okay? I learned very quickly the talk to the hand response, right? So the idea is that the immune cell would come up ready to, go after the cancer cell, deliver a lethal blow, and the cancer would hold up this thing, say, look, talk to the hand, go somewhere else. And that response was this discussion, if you will, between PD-L1 and PD-1 and these kind of things. That response could be interfered with by an antibody drug against, by the way, either PD-1 or PD-L1. It's the recognition sequence protein on the surface of the immune cell and on the cancer cell that together creates the talk to the hand response. And so when those antibody drugs stop that conversation, then the immune cell is unleashed to go attack the cancer cell. And in about 15% of human cancers, it works incredibly well. 40% or more of melanoma. You heard that President Carter had malignant melanoma with metastasis to his brain. This is what he was treated with. It completely disappeared. He's 100 years old now in hospice care, but his cancer is still completely gone years later. This is revolutionizing the treatment of lung cancer, bladder cancer, and several others. The other thing you've heard a lot about, and this will make a little bit of sense to you, is that the other thing you can do is you can take immune cells outside the body, the ones that can kill things like cancer cells, and you can insert genes in them so that they recognize all of the ones you take out of the body. They all recognize the cancer. It's sort of the, it's like raising the infantry, 10 to 1,000 guys who can go in and chase down the cancer. And you can reinfuse them and they'll chase after the cancer. And these are called chimeric antigen. That means you've changed the way they recognize things. T cells, that's the kind of immune cell they are, CAR T cells. And these are a tremendous tool to use to treat leukemia in children, to treat certain lymphomas, to treat myeloma. And they've had a little bit more troubles thus far chasing after breast cancer and prostate cancer and the like. And there's a lot of research in the area to figure out why doesn't the talk to the hand antibodies work in these cancers? Why doesn't the CAR T cells work in these cancers? And I think they're getting kind of a bead on it. It's almost like an ecological problem. The microenvironment around the cancer is completely polluted. The cancer cell eats all the food, and it deposits as waste ammonia and acid and these kinds of things. And it's a tough environment for the immune system to work in. So I think if we can get around that problem, we'll then be able to eradicate even more cancers than we already do. So if you look at how fast this is going back in the 1890s, these Coley's toxin, witch's brew kind of stuff, but look how quickly things picked up after 2010. You're starting to get antibody therapy, the immune checkpoint inhibitors, that's the talk to the hand kind of antibodies, other kind of antibodies. And the latest thing that's out there now is to take the sequence of all the defective proteins, all the defective genes, all the defective proteins in the cell, use them to stitch together an antibody that's very specific for all my defective genes in my cancer versus someone else's. They'll be different, and then use them as a vaccine along with some of these other immune tricks. And there have been some promising results from that just in the last half of a year or so. That's sort of where the field has moved at this point. So where are we at with all this? I told you that there's this incredible energy in DNA-based diagnostics, which may transform the way we do screening and early detection. In the design of small molecule drugs, we can tuck them into these little protein mini machines and hopefully stop the defective cancer proteins from working. We can build antibodies that recognize the cancer cell, load them up with payloads so they can kill cancer cells and do very little in the way of other damage. And we finally have the immune system in play at least about 15% of the time or more. So where are we at now? This is a little report card I did for another purpose, and 25-year anniversary, actually, of the V Foundation, the ESPN charity. And if you look at the death rate has fallen substantially. The 5-year survival rate is still now greater than 2/3 of folks, and we're walking our way towards 4 million fewer deaths than expected. And you can see this in some of the common cancers, right? The 5-year survival for breast cancer, for prostate cancer, colorectal cancer, it's significantly improved. Prostate cancer is an interesting one. If you look at a man diagnosed with prostate cancer today, he's got a 99% chance of living 5 years. That's actually better than a man of the same age with no disease of any kind. Part of that's a quirk of statistics, but it's an interesting one. There's things that we know we have to do better at. Pancreatic cancer and liver cancer are on the rise throughout the country and in Maryland. We don't do well enough against these cancers. It's a lot of effort now, and it's been hard. It's been a hard slog. We haven't had the kind of benefits against those cancers that we've had against many others. Leukemia, we're starting to do better. Ovarian cancer is sort of a work in progress, just starting to improve. So we've got a long way to go, but we've taken some giant steps, and you can see how the future is going to unfold. You can see where artificial intelligence is going to take us. We're going to make better drugs. We're going to make better predictions about who needs which treatment. It's all going to be individualized, and hopefully we'll see a time when people aren't so threatened by the notion of you have cancer. So thank you. So I know Jayne is going to manage the questions.
Okay, thank you, Dr. Nelson. Before we take some questions from the folks who've been listening on the webinar, you spoke about the importance of tumor sequencing to personalized treatment, immunotherapy, liquid biopsies, new medications, and proton beam therapy. How are you accelerating research for those new treatments?
Yeah, there's nothing like talking to a reporter asking you questions. Yeah. So what's happening? If you look at the what you see here in the academic medical centers like Johns Hopkins, and particularly University of Maryland, somewhat as well, is you see people doing the initial discoveries. What are the targets that we can aim drugs at? What exactly is going on that turns something into a cancer in a very basic level? What are the new platforms we might use to detect DNA and the defects? And as these things become clearer and better understood, some of them will deliver new ideas, new intellectual property, if you will, that are the basis for the new products that get out there for early detection and the drug treatment and whatnot. So I think what we try and do at an academic institute is we know a lot about the diseases, so we try and discover how the cancers work, what the defective genes are doing, what we need to stop them with, and the back end, when products start to appear, we see the patients, we understand the disease, we try and figure out how best to use them to create benefit.
Okay, this is a question from the attendees. What about colonoscopies for people who find the whole process uncomfortable? Haven't we all been through that? And how effective are the blood tests and Cologuard? Are they a good alternative to diagnosing cancer disease?
Yeah, so some of these stool tests are pretty impressive. Cologuard's a reasonable one. The secret to the Cologuard test, yes, there's a little bit of a DNA test in it. The other thing they're testing for is human blood. They have a very good antibody test for human blood. There are several of these kind of tests out there in addition that aren't the premium price that Cologuard is either. And they're pretty good. And in fact, when you start looking at colonoscopy, the advantage of colonoscopy, if you have an expert that does it and they withdraw it slowly enough that they can see everything on the way out, which is how they do it, that if they see a benign kind of lesion, even though it has a low chance of going to be a cancer, they can still remove it. And so they can biopsy things if they see something abnormal. If it's a pre-cancerous kind of thing, they can remove it even if it's a reasonably low risk one. So you can get all that done in colonoscopy. Having said that, the number of folks that can have access to colonoscopy, particularly in rural parts of the country, are very limited. So if we're going to actually control colon cancer, we need to make maximum use of all these tools. And so these ones that you do stool testing, obviously you can deploy that in a far greater range of folks across the country. And if the test is positive, then you can figure out a way to get colonoscopy if it's a little bit scarcer resource, if you see what I mean.
You published an article claiming that 60% of cancer results from just bad luck. What are the lifestyle risk factors, including alcohol? I think there's been a lot of research recently about that that can influence.
Yeah, I did try to write something to make sense out of that. There's a couple of great overall logic for where cancer comes from, right? So the genetics, how much of cancer or risk to get a cancer is inherited. Well, there's the famous kind of studies that look at twins. Identical twins share all their genes. Fraternal twins, they call dizygotic twins in my business, share half their genes. And yet most of the time they grow up in the same house, they eat roughly the same food, exposed to the same thing. So if there's a difference in the concordance of a cancer diagnosis between two identical twins versus two fraternal twins, the thought is that that's genetics, not so much the environment. So when you look at that, there's a significant fraction of cancer attributable to genetics, probably on the order of 40%, which is a substantial fraction if you think about it. The environment, there's a great classic set of experiments, geographic epidemiology. The most famous one is that historically there was a very low risk of breast cancer, prostate cancer, and colorectal cancer in China. Of course, that's prostate cancer in men, breast cancer in women, but a very high chance of liver cancer and stomach cancer. If you look at Japanese immigrants to North America, their risk of stomach cancer and liver cancer falls, their risk of breast, prostate, and colon cancer goes up. This is without intermarriage. So this is the same genomes, but in a different environment. So when you guesstimate that effect, most people think it's between 85 and 90% of all cancers can be attributed to some kind of environmental exposure or lifestyle habit. And then the bad luck idea was that somebody pointed out that every time one cell becomes two and they need to copy those 3 billion base pairs, they make a lot of mistakes. Most of them are fixed, but there's about 7 to 10 each time one cell becomes two that aren't fixed. They're just too hard to fix them all, I guess. And so they accumulate over time. And the thought is that if one of those happens to be in exactly the wrong place, it's like the reverse of winning a lotto ticket, they might affect the cancer gene, in which case that had nothing to do with your genetics or the environment. It just was, one of the places where there was a mistake made during the copying. And the logic there was that cells that copy themselves more should have more likely to be a cancer, and that's where they got this started. They estimated that that was 65% of all cancers. Now, I'm a higher order math kind of guy. When I add 40 and 85 and 65, I don't get 100. Okay, so they clearly have to all interact. And some pretty good evidence that even with very strong genetic predisposition to cancer, Mary Claire King, one of the people responsible for discovering BRCA1, pointed out that there's pretty good evidence that there's a significant lifestyle effect on the age distribution of when women with BRCA1 mutations, which is a strong genetic risk factor for breast and ovarian cancer. There's still some lifestyle effect even in that setting. So yes, just what you said, you want to reduce the amount of alcohol, you never want to smoke. It looks like tobacco use is curtailed enough in this country, so the dominant environmental risk factor is probably, some people call it a three-legged stool: eating too many calories, not doing enough physical activity, and storing your calories in an incredibly inconvenient way so that you get metabolic syndrome and the like. And that seems to increase risk for many of the cancers that we get in this part of the world. So.
Okay, well, then this leads to the next question about what you eat and the connection to cancer. What is the research about that? I think you mentioned some of it, connecting. This is about the microbiome, et cetera, and the gut and the human body, et cetera. But this really gets to, what you can do individually in terms of your nutrition and diet.
Yeah, no, this is a tough one. You look at how are these inferences generated, and you can imagine that you take a group of people, you give them questionnaires, and this is a tough business. I mean, if I ask you, what you had to eat for lunch 3 weeks ago last Tuesday, and you could answer that question, you don't belong in a study of normal people, right? So the notion is they ask you, how often per week did you eat this particular food and that particular food? And they've done a lot of work to optimize these, so they're pretty good, right? Then you take people who have a disease, in this case, a cancer or didn't have cancer, and you say, are there any sort of differences in what was consumed. There are some ways to make this work better. It's better if they get the questions asked before they knew they had any kind of cancer. So you get them enrolled in a study, and later maybe they get a cancer. You go back and say, what were they think they were eating? And anyway, from this, it's the same kind of risk factors you see for cardiovascular disease. If you a diet low in fruits and vegetables, high in processed foods, red meats, saturated fats and the like, tends to be more prone to develop cardiovascular disease and cancer than not. When you get into the details of it, it gets quite interesting. There are a whole bunch of leads that may get clearer and clearer over time. One of them is cooking practices, right? So if you take meats and you heat them to very high temperatures, you create in them, by a heat catalyzed sort of reaction, you create these heterocyclic amines, are called, particular chemicals. They're interesting. If you take them and feed them to laboratory rats, a Japanese laboratory did this, the male rats get prostate cancer and colon cancer, the female rats get breast cancer and colon cancer, and that's just plain interesting for something that you eat. You make a lot of these things when you pan fry things, when you heat things to high temperatures. You make the same kind of things when you light a tobacco leaf on fire, only this case you're eating them instead of smoking them. And there's some estimates that humans in this part of the world maybe eat as much as a pack or a pack and a half worth of cigarette smoke a day if they eat these products. That's one that there's a lot, the jury's not fully in on, but it's one that you can probably avoid. You can steam foods, you can bake them. Anything that avoids pan frying probably helps with that. There hasn't been anything that's been untoward about eating fruits and vegetables. So
this is a question from one of the participants. You mentioned finding the proper treatment dosage, but how does duration of treatment affect cancer outcomes? And does having prolonged chemo treatments have significant negative effects?
Take the second one first because it's easier. Yes, having a lot of chemotherapy, particularly, we started learning a lot when we began curing people, because then they live long enough to have late effects, not just the side effects you see early on, where from chemotherapy you might lose your hair, your blood counts fall and rise again. You might have difficulty with numbness and tingling in fingers and toes. When you look long, there were certain of the chemotherapy drugs that tended to cause leukemia, and people that got cured of Hodgkin's disease back in the 60s and 70s. We don't really use those chemotherapy drugs anymore for that reason. There are other drugs have risks of other problems almost all throughout the body. And I think the answer is, when you can, you minimize the exposure to them. If you look at testicular cancer now, someone gets adjuvant therapy, it's now only one cycle of treatment. It's not, a year's worth or anything, just one cycle. So I think you want to minimize the treatment when you can. You want to reduce the propensity for some people to need the treatment, be a little bit clear on who needs treatment and who doesn't. But yes, if you lean on a lot of chemotherapy, lots of different cycles, there are untoward proximate fatigue, brain fog, these kinds of things, and then later effects that aren't attractive. So
I have a question about breast cancer. Are there better drugs on the near horizon than inhibitors for breast cancer?
By the inhibitors, they probably mean the hormone receptor antagonists. There are some on the horizon. One of the most interesting ones is the antibody drug conjugate against HER2. There was one that had come out a year or two ago, antibody to the HER2 protein, which is 10-15% of breast cancer. And this one was linked to a drug, and it was pretty effective. And people are beginning to wonder, could you get by with just something like that in certain circumstances? The next one that has come out is a better drug that is, it threads the needle better. It swims around on the antibody, so you don't get exposed to it, and it gets clipped off right in the cancer pretty effectively. And it's extremely effective, and it'll be interesting to see how many of these things we can build and get a hold of for breast cancer. I think if you can get breast cancer cured in such a way not to necessarily interfere with estrogen action, and a younger woman, making that younger woman premenopausal, sending her through a menopause at a younger age has other untoward effects, right? Loss in bone mass and these kind of things that are less desirable.
Actually, the question about what younger folks can do to improve their health outcomes.
Yeah, that's actually a very pressing question. There's a big concern just in the last about 5 to 10 years that colorectal cancer was the sort of the canary, if you will, in the coal mine, where we started to see across the country younger people getting colon cancer. Usually we were recommending that people start getting screened, colonoscopy or one of these stool tests at age 50, unless there was a family history or some other reason to start earlier. We're now seeing colorectal cancer a lot more commonly than we should be seeing it, and a lot more commonly than we used to see it in younger people. And so already there's a drive to start screening at an earlier age. Most people believe that is related a little bit to the thing we were talking about before, this too many calories in, not enough physical activity, and storing fat inconveniently, sort of the obesity epidemic. So keeping a normal weight, keeping yourself active, eating a prudent diet that doesn't have so much concentrated sugars and stuff that you get obese. In other words, go after those fruits and vegetables instead of that kind of food.
What
That's
efforts
a
have been
significant
made?
worry young
I'm
people.
sorry.
That's a significant worry young people cancer. It's kind of
Well, I know, and I've heard others actually from Hopkins talk about the increase in the incidence of colon cancer in younger people.
Yeah, it's a real thing, and people are really trying to chase after that.
What efforts have been made in increasing efficiency in drug targeting?
Yeah, that's a great question. Yeah, I think the key to the efficiency, in other words, what you really want to do is very selectively hit your target, and if ideally hit absolutely nothing else. So the only effect that you have is the effect of hitting that target. Now, you might imagine a side effect if that target is somewhere else in your body, then that would be an on-target side effect. You can't avoid that. But if it really does tuck itself into one of these defective proteins that are only in the cancer, then ideally you should have no effect anywhere else. And I think that's the goal of a lot of the artificial intelligence-driven sort of recursive small molecule drug design that I splashed at you in that one particular slide. What's interesting about that is that these drugs are getting increasingly more potent. Potent in a pharmacologic sense means less of the drug is needed to have the effect, and they're getting incredibly potent. They're also increasingly pretty stable, so that you can take the drug as a pill, you can absorb it easily to some degree, and then you can excrete it in the urine or in the stool or something, largely unchanged. It doesn't affect it as much ideally by metabolism that might be different in you than me, so the drug becomes more predictable. One of the challenges with that is if they're that potent and they end up leaving your body in an intact form, then as they get into the sewer system, they start to do things like affect the fish and stuff. So it's an interesting problem. The drugs are getting very, very potent.
Is there anything new in treatment of pancreatic cancer?
Yeah, there is, and it's providing a little bit of hope. There was one of the first of the personalized vaccine kind of thing, where they take the pancreatic cancer, they sequence the genome, the tumor DNA in the cancer, they get a whole inventory of all the defective genes. They use that to plot which of these defective genes can they use to stitch together into a vaccine. The vaccine platform they used was actually the mRNA vaccine platform, because they can make it quickly and then administer it to people after they've had surgery, and they're also getting some chemotherapy. In that group, the people who were vaccinated, and you could test them and see what their immune system was up to and clearly see a vaccine response, they definitely did better than the people who didn't have a vaccine response. And the majority of them did pretty well. So it looks like an incredible first step. It already, even within that study, suggests that there's more work to do, but there are several of these kinds of approaches out there now. They do seem pretty promising.
You have spoken a little bit about this, but what about ways to block genes from metastasizing?
Block the cancer cells from metastasizing? Yeah. So there are a number of approaches to do that that seem to work in, curing mice is an exciting business, but not as fun as helping out people. A lot of times there are ways to stop something from metastasizing when it clearly hasn't yet. The challenge then is that what's the opportunity to really test that clinically? When would you use that? In other words, sometimes we worry that when someone has a cancer and they ultimately suffer with metastasis, they already had metastasis by the time we found them. So perhaps these drugs that interfere with the process of metastasis, and there are some that look like they do that, will be more and more useful as we get to more and more early detection, if you see what I mean. But it's been hard to use a drug like that now in a setting where someone doesn't already have metastasis.
Can you expand on information that's been released by Johns Hopkins showing that immunotherapy before and after lung cancer surgery and the relationship to reducing death risk and recurrence?
Yeah, so this is one of the places where this they call immune checkpoint inhibitors, that talk to the hand response that I was giving. This is one of the places where those antibody drugs that do that have had the greatest success. Probably the greatest success has been melanoma. The next best success has been in lung cancer. And you can use these drugs before the surgery. The clinical trials were done that way in part to see how they work, but also because then when at the time of surgery, you can see what the immune system was doing, specifically what was it doing in the area around any cancer that was still there. And the treatment was effective on its own. It's now effective when given along with chemotherapy, and it's become the standard of care. It definitely has changed the game for lung cancer. Not enough people with lung cancer, but for a lot of them.
I think you've also kind of addressed this a little bit, but what research is being done in identifying key markers in various cancers, for example, pancreatic cancer, tumor markers for various cancers?
Yeah, the big thing that's happening now is there's been a technological advance that enables you to get a cancer tissue and then disaggregate it into individual cells and get a sequence of all the working copies. That's the RNA. So the DNA is the blueprints. It's made into RNA, which are the genes that are being used, and they're taken to the site where the proteins are produced. You can literally sequence every RNA in just one cell. And so you do this for every cell in there, and you can literally tell, I'm trying to think of the right, metaphor or analogy. You can tell every person that's there, and you can tell exactly what they're wearing that day. And so you really see what the entire microenvironment looks like. And you begin to see what the microenvironment looks like when the cancer is successfully treated, what the microenvironment looks like when it's not successfully treated. Are there cells in the microenvironment that there's a pretty good hint that there's one kind of immune cell that shows up that makes it very hard for the immune system to kill the cancer. And there's already a bunch of strategies to try and get rid of that cell from the microenvironment and unleash the immune system. So this kind of analysis where you look at every individual cell, some of them are cancer cells, some of them are support structure cells, they're corrupted as well. Some of them are immune cells, some of them are immune cells that are taking the day off, they're not doing anything. And you can really get a look at all of them. And that lots of computational energy and significant technology to do that. And everybody's doing that kind of analysis now at all the cancers that we treat.
I just want to go back one last time to food, because we do have a question about what about frying foods? You talked about, heating meat to a very high temperature, which of course would probably mean grilling. But what about frying foods in terms of its relationship to, a lifestyle risk?
So there's the pan frying. One is just caused by heat alone, one of these sets of compounds. The other is caused by actually burning them, which is the charbroiling, it's called. Pan frying, the thing that's complicated is there was a great little study of this where they took it tells you what the answer is, by the way, too. They took it's identical equivalent burger patties, and in one of them they cooked it at a high temperature and left it on one side for like 5 minutes and then flipped it over for 5 minutes. The other one they had on a slightly lower temperature and flipped it repeatedly. And in each case, they then measured any E. coli or anything that's in the meat. Was it adequately killed? And in both cases, it was. But in the one where they fried it intensely and then flipped it over, there was 10, 50-fold more of this compound. So you can avoid it, it turns out these compounds if you pan fry, not as high of a temperature, flip it over, marinating actually gets in between the compounds that get heated. That helps. Baking, steaming, all these kinds of things turn out to work pretty well, a little bit better. So you don't have to give up on the food, just probably that the salt crust that tastes so good on the steak or the burger may actually be the problem. So,
But just preparing it differently.
yeah, the one that got me is that there's used to show a slide to the medical students, see if they recognize. There's a picture of collard greens, right? And collard greens should be a superfood, right? I mean, they're incredible, like kale and whatnot. But if you look at the classic way collard greens are cooked in the South, they're usually cooked in the pan drippings of typically a pork product, something that was cooked in the pan before. And the reason you do that, of course, is it tastes much better when you do that. But that's almost a concentrated extract of these things. So here's a healthy food that's made unhealthy by the way we prepare it.
Last question has to do with affordability of treatment. Has there been progress made in making treatment more affordable?
No, I mean, not anywhere near what it should be. This is a major issue, and I think it's going to play out in the coming year. That there's as part of the act that President Biden did, there's a propensity for Medicare to do more bargaining for the price of the drugs, and the large pharmaceutical companies are upset. Many of them are suing the federal government. I would be more sympathetic, actually, like the biopharma industry. I love the idea that we can make new drugs that can help people, but they spend a lot of money on direct-to-consumer advertising. I don't put my political stripes on. I can't believe they do that. I don't know why they do that. I just don't think they should do that. Spend that kind of money. Spend the money on research and development. Build new drugs and better products, and everyone will think more highly of you. There hasn't been that much market competition for these drugs yet either. There are now several of these immune checkpoint inhibitors. The price hasn't gone down very much for several. So I think that the pricing is a problem. But I think that they're grappling with it at the policy level, and we'll have to see what happens. So.
Okay, as we approach the end of tonight's webinar, I'd like to thank Dr. Nelson for speaking with us tonight. In a couple of weeks, you'll receive an email with answers to any outstanding questions and answers that you might have. A video of tonight's live stream will be available on the Woman's Journey website. Again, that's hopkinsmedicine.org/A Woman's Journey under videos on demand. If you have enjoyed tonight's discussion, please check out the website, hopkinsmedicine.org/A Woman's Journey, for more information about upcoming live programs, webinars, podcasts, and the monthly email. And especially read about the forthcoming program, A Woman's Journey, on the morning of Saturday, November 23rd in Baltimore. It's a great event. You don't want to miss that morning event with 10 presentations by Johns Hopkins physicians. Good night, everybody, and stay well.